Today I will be sharing my talk about drug discovery using symbiotic microorganisms from Panama. Okay, I divided my talk in 5 parts. First, I will talk a little bit about symbiosis and natural products, and then I will talk about different symbiotic systems under the study at INDICASAT in our research group. And one of the systems is the frogs, and then fungal growing ants, and then corals. Okay, some some scientists start to seeing this kind of similarities in the nature where you find two compounds that have same structural nucleus like onamide A and pederine, like very similar compounds produced by so distant organisms like a sponge and an insect. So in the case of onamide A, this compound is cytotoxic, and in the case of pederine, this is a defensive compound that produces a huge pain in the people or any animal that interact with this insect. So this brings speculation about the real biosynthetic origin of this compound. That very likely is not the sponge or the insect, the real producers, but are the microorganisms that live associated to these microorganisms that are the real producers of these compounds. And then in 2014, the group of John Peel from Germany actually discovered that onamide A is really produced by a symbiotic bacteria associated to the sponge of the genus Theonella. So confirming the real biosynthetic origin of onamide A and pederine. So also in the case of Jondalis, Probactidine, that was first isolated from a tunicate, and it is active or is used to treat soft tissue sarcoma and ovarian cancer. So these compounds also have a nucleus that is very similar to some bacterial metabolites. We can see here different genus of bacteria produce the same nucleus. So and actually in 2011, the group of David Sherman at the University of Michigan demonstrated that the real producer of Jondalis is bacteria that live in association with the tunicate. Okay, so here there is a table published by Bill Gerwig and collaborators, and you can see in 2012 you can see an update in these marine drugs for this year. So here we can see several compounds that are approved and in the market already. And this is the organism from they were first isolated. And then this is predicted by a biosynthetic source. And in some cases, the real microbial origin is demonstrated, as I show you. And you can see here the therapeutic area. So there is a trend to see that some of the drugs in the market that were produced by microorganisms like marine invertebrates or plants, actually the real producers are microbes. Here there is an example. The Taxol is a famous compound used to treat different kind of cancer, and it was first isolated from the tree Taxol brevifolia. And now we know that Taxol is actually produced by fungi from different kinds, isolated from different kind of trees. So there is a real trend to find that drugs in the market are of microbial origins instead, that coming from the microorganisms. Okay, so given this trend, we decided to study different symbiotic systems that we have available here in Panama. And one is in the marine environment with octocorals and sponges, and also the fungus-growing ants and the frogs. So I will talk about these three projects, and the first project was carried out by one of our graduate students, now is a doctor, Christian Martin. So he worked in the frog systems, and as all of you know the frogs and amphibians around the world suffer like a catastrophic declines due to the attack of chytrid fungi. You know, and in many places this produces like complete extinction of many amphibian species. So some scientists found that some amphibians were resistant to this disease, and they found that these amphibians have bacteria that inhibit the growing of the chytrid fungi, as you can see here. So we decided to take a look at the amphibians from Panama to see if we observe the same pattern. And we make a little search, and we find that only 4 compounds at that time were published with activity against the chytrid fungi. This is like a complete unexplored area to study. So we collected 8 frog species from different locations in Panama, and we isolated 2001 isolates of bacteria, of which 29 were active in an assay against Aspergillus fumigator. And we can see here all the strains that were active, and we can see that Pseudomonas chicory showed the greatest activity, even more active than the control cyclohexamide. Okay, here is the frog, and here you can see the Pseudomonas that is inhibiting the growth of Aspergillus. We can see the inhibition halo that indicates the presence of microbial metabolites. Okay, we study this interaction using a technique called multi-IMS. This multi-imaging is mass spectrometer-based technique. So in this technique, you can use the multi-laser to scan this area where there is the inhibition zone, and then you can give a different color to all the metabolites that are present in this inhibition zone. So here we can see in the interaction, this is the control, the bacteria and the fungi, and here is the interaction. And we see different color for the different compounds that we see as we're using multi-EMS, so we can know the molecular weight or the masses of these compounds and give a different color. So this gives you an idea of what is the metabolite that we have in the interaction zone. So we make an extract and then of this interaction, and then we study this extract using GMPS molecular networking, which is a technique based on tandem mass spectrometry. And using this technique, you can identify entire family of compounds. So this analysis produces this kind of clusters, and if you identify one of the compounds, then all the nodes that you see here that cluster together belongs to the same molecular family. And using this approach, we identify viscosine and the macetolide are analogs, lipopeptides that were present in the inhibition zone. So we replicated these compounds using GMPS molecular networking. You can see the paper here, the scientific report 2019. You can see more details on this technique. And so in order to confirm the structure, we make a MSMS analysis of all these peptides, and we confirm the structures using mass spectrometry. to know the sequence of different amino acids that form the peptide, and also to prove the concept, we isolated pure viscosine using chromatography and confirming the identification using NMR. So actually, this was confirmed. The structure of all these compounds were confirmed by isolation and use of NMR mass spectrometry. So, as we isolated the pure viscosine, we evaluated viscosine and the assay against Aspergillus fumigatus. And as we can see here, we see that viscosine produced inhibition against Aspergillus fumigatus. Also, we tested viscosine against the chytrid fungi that produce the amphibian disease, and we found that viscosine was also inhibiting the growth of the chytrid fungi. So this confirmed that the frogs maintain bacteria that are protecting them against the attack of these chytrid fungi. Okay, so we're going to the second project is on fungus growing ants. It was carried out by Dr. Christopher Boya, also from our laboratory. And this is about ants that keep some actinomyces. We have a thin ant here, and this black powder that you see here is an actinobacteria that they keep. These are farmer ants that maintain fungi in their nest, and then they feed the fungi with the ants, with the leaves. Sorry. So they are kind of farmers' ants. So as any farmer activity have like plagues, and in this case there is a fungal pathogen that attack the farms of the ants, producing the decline of colonies. So some people found that this actinobacteria that the ants keep actually inhibits the growing of these fungi, and they use this bacteria like a kind of a biological control, from million years ago of evolution. Okay, we use the same approach, studying the interaction zones using multi-imaging. We can see the actinomyces bacteria, and we can see here the Scobopsis fungi, and we see the clear inhibition through the production of metabolites. And here we can see different colors, the presence of different metabolites. So we use also molecular networking, and we found lyophilin and ephromycin G and A present in the molecular networking. So to prove the concept, we use chromatography to isolate pure lyophilin, and then we confirm the structure using NMR, and also we tested the biological activity, and this compound show antibacterial activity. Very likely because the ants use this compound to keep control on bacterial communities in the nest. And also this compound showed antimalarial activity. Okay, we also isolated some compounds from the fungi, Scobopsis, in the inhibition zone. So the sharing in F, D, and J, and this was the first report on the occurrence of these compounds in this kind of system. It was published in Scientific Report 2017. Okay, and my last example was carried out by Dr. Librada Atencio from our research group also. And Librada was dedicated to study some marine bacteria that were isolated from different marine vertebrates like sponges and corals, tunicates. and she focused on pseudotheromonas. As you can see in this phylogeny, this part here are non-pigmented species, and in this part of the dendrogram, we can see pigmented species, and this is the antimicrobial profile. Here we can see the different targets, and we can see that the pigmented species show a really, really great antimicrobial profile. So we decided to study one of these, OT59. We isolated the bacteria from Leptoborgia alba and octoporal, and we found that these bacteria inhibit a fungi that was isolated from an ear coral. As you can see here, there is a clear inhibition zone produced by the bacteria. So, but we observed like a real phenotype, and in the continuous light, there were no inhibition. You can see here. So, in a day-night cycle, we observed some inhibition, and in continuous dark, we observed continuous inhibition. So, it was kind of very rare. So, we studied it using also multi-imaging. As you can see here in the dark, there is the inhibition and the presence of metabolites here. You can see different colors, different masses of the compound. And in dark light, the activity is not that big, and then in the light, there's no activity. So we identified the replicated this using mass spectrometry. You know, we have the molecular weight. We found that they were alteramides analogs. And also we found some bromoalterochromide that was not affected for these day-night cycles. So in the case of alteramides, we made the complete isolation and identification of the compound using NMR. We found the alteramide A, which is has antifungal activity. When you in the presence of light, you suffer kind of internal cycloadditions, and you can see this is product here, which is not active against fungi. This is active, and this is not active, and this was this is the explanation for this light-dependent bioactivity. Okay, we selected another species of pseudotheromonas also to study, and then we used like a genomic approach, and we found that these species have the biosynthetic gene cluster of bromoalterochromide. So we used the pseudotheromonas PCC that was published in the gene bank, like example, and we found similar clusters that produce bromoalterochromides in our species. Using molecular networking, we found a family of known bromoalterochromides. They match perfectly in the same cluster, and we also found 2 new compounds using this approach. I want to mention that for this study, we only use extract and mass spectrometry to identify the molecule. We never use chromatography to, to obtain the pure compounds. So LCMS for the analysis of mass spectrometry, analysis of this compound. So we confirmed the identity using MSMS tandem, and then you can see the structure of the 2 new compounds that were published in Marine Drops. So, so this is the show that this technique is a powerful technique. Mass spectrometry is a powerful technique to replicate compounds and also identify compounds, even without making separations or obtaining pure compounds. Okay, my conclusions, our results confirm that symbiotic microorganisms from Panama, beyond their intrinsic value, have a great capacity to produce molecules with potential in drug discovery. and also the integration of new analytical tools for metabolic analysis and genomic tools will impact the drug discovery process. So, for the question of this section, is bioprospecting the way? So I would say no. If we keep using the same traditional approaches, so if we use traditional approaches, we'll continue isolating the same compounds. But if we use the new tools that we have available, like genomics and this new analytical capacity-based mass spectrometry and NMR, so then we will find, a universe of molecules, new molecules actually. And to prove my big guess, so I found this paper published by the group of John Peel very recently, Nature Review Drug Discovery. You can look at the reference, and they demonstrate that the bacterial diversity far exceeds what has been previously observed by classical cultivation. Also, that genome analysis has revealed a tremendous diversity of biosynthetic gene clusters that far exceeds the number of known substances. So, through using these genomic and analytical tools, it's demonstrated that we only have studied a very small portion of the natural products that are present in nature, and probably using these new approaches, in genomics and analytical tools, we will find new chemistry that is, waiting for being discovered. And so they estimate that very likely next years, or maybe in the next decades, so we will see new compounds entering in the market using these approaches. So I have to say thanks to the research group. Christopher is the person who worked with the ants. Here is Librada, who worked with the corals, bacteria, and Candelario. And Christian, that worked with the frogs. So I'll be happy to answer any question. Thank you.
Okay, Marcelino, thank you very much for the presentation. Very exciting information. I find this symbiosis certainly playing a very important role in secondary metabolism pathways, which I wasn't very aware of. And thank you for sharing all this information. And, I was thinking, we know that it's always hard to go through the whole process of drug discovery in countries like ours, for instance, in Latin America, not only when it comes to research and secondary metabolites, but in general. More often than not, we end up being suppliers of genetic resources. But that does not seem to be the case in Panama, specifically with Indicasat, who are managing to do most of the research in-house. How did you achieve this in Panama? What do you think that this is? What do you think that still has to be done? And I've always been impressed by this in Panama. Always very strong culture and experience in natural products research.
Well, it's been a process. I remember when I was a bachelor student, we even didn't have any more mass spectrometry here in Panama. So it's been a process. First, I think with projects like ICBG that was funded by NIH, and also project continue the funding like projects like GEF. We're able to build the capacity, and also with the support of the Senate in Panama, also building the capacity, building laboratories, buying equipment, and then, it takes many years to have all these things. to make research competitive internationally. And I think it's been a long process. It's not easy because we have, in Panama, we have problems with the maintenance of the equipment. It's not like being in the US or in Europe. You know, here it's more difficult to get, the humidity in Panama is huge and this is bad for the equipment. So there are challenges, but at least we have some equipment and some, infrastructure built by the government of Panama and also projects like ICD and GAP. But it's been a long process, and also a lot of international collaboration. With people that have the expertise. We send students to learn and then come back and apply of these methodologies here in Panama.
Okay, okay. Congratulations for your team and your country. And well, thank you very much, Marcelino.